In the last lecture, we looked at what are amino acids and their properties. Let's now discuss the structure of proteins. Proteins can be subdivided into four based on structure: primary, secondary, tertiary, and quaternary structures. Primary structure is also referred to as linear structure. On the right hand side, you can see primary structure of protein, where amino acids are arranged next to one another through amide linkages. So the amide linkage on the left hand side, you can see that it is formed by condensation of alpha carboxyl group of one amino acid with alpha amino group of another amino acid. They interact, they undergo condensation reaction to form amide or peptide bond. Now, during this phenomena, there is loss of water. Amino acids have L configuration in this linear structure. So what does it mean? The main substituent group, which in this case is amide group, is present on the left hand side of the chiral carbon atom. Proteins, they also tend to have a free N-terminal amino group and a free C-terminal carboxyl group, which represent the beginning and end of a polypeptide chain. Now, the physicochemical properties, structural properties, biological properties, and functional properties, all of these things, they are determined by the chain length of the polypeptide that is formed, and also the manner in which these amino acids are arranged in the polypeptide chain. The amino acids, they also carry code which is necessary for the formation of secondary and tertiary structure of your protein. One important feature about the amide bond or the peptide bond that is formed is it is a partial double bond. So what is meant by partial double bond? I'll try to explain it. So the nitrogen present in the amide, it has the capability to keep the amide bond stable by transferring its lone pair of electron to the carbonyl carbon present here. Now from this carbon, it goes to oxygen, probably attributed to the high electronegativity of oxygen. It always has a tendency to attract electron towards itself. So this partial double bond character, it has several implications in terms of functional properties of proteins. First is that amide group cannot be protonated. Second, because of this, the amide bond that is formed has limited rotational freedom. What does it mean? It means that the flexibility of these bonds, they are very limited, almost limited to an angle of 6 degrees. Okay, you do have few angles which have some sort of rotational freedom, that is your phi angle and your psi angle. The third is that because of electronegative nature of oxygen, okay, and transfer of lone pair of electrons to oxygen, there is possibility of hydrogen bond formation within the protein structure. But for simplicity's sake, what we do is that we consider amide bond as a single covalent bond. Thank you for staying with me through the video. In case you have any queries, please feel free to put them in the comment section and I'll get back at the earliest.